Working properly. Check my speaker. Hello, testing. Okay.
Greg, I think we've only met once. It was at a study section in Washington before COVID. I think
could be, yeah.
I don't think I've ever met you, Greg. Hi, I'm Anna. I'm the admin for IBANGS.
Hi, Anna. Nice to meet you.
Nice to meet you. So, let's see. Here's Rita. Rita's joining. Maybe she wants to test some slides. Let's see. How's the video now, Paul? What do you think?
Your video is pretty choppy. I can hear most of what you're saying, but
okay. I'm going to turn it off soon. I just want to say hi to Rita and say hi to some people. Hi, Rita.
Okay.
Hi.
Hello.
My name's Anna. I'm the admin for IBANGS. It's great to meet you, and thank you for giving a seminar. We're excited about your talk.
Thank you for inviting me.
All right.
Hi, Rita. I'm going to give you a brief introduction before you speak. Very brief, like 2 or 3 minutes. Okay. If I'm going to pronounce your last name, do I pronounce it Cevera Juanes?
Perfect.
Am I embarrassing myself if I
you're not.
Spanish accent?
One of the best ones I've heard.
Okay. When I graduated from college, I spent about 2 weeks in a small town called Piles outside of Valencia on the coast. Just hanging out and doing the postgraduate thing, and I love that part of the country where you're from.
Thank you. I do too.
And now you're in one of my favorite parts of this country. I got my PhD at OHSU, and I just love Portland. Yeah, with Tamara Phillips.
Oh, okay. Yeah. Of course. Yeah. Small world.
So we've traveled in the same circles, but at different times.
It's a nice coincidence. Now we're back meeting each other.
Right. So Anna, you're admitting all the people too. I've been hitting the button too. If you're doing that, then I'll stop. I don't want to mess anything up. I think I'll just wait maybe in 1 or 2 minutes, and then I'll introduce you, Rita, and then we get started.
Do I go ahead and share my screen now, or I wait?
Yeah, it'll probably be good just to practice it. I mean, just to make sure that you can share.
Is that good?
That looks great. We see the full screen. It's perfect. Well, I guess this would be a good time to start. Why not? It's 12:01. So hello, everyone. Thank you for joining us. This is our first IBANGS seminar of this academic year, and I'm really excited that all of you have turned out because we got a really good show today. Seriously, I'm really excited that Dr. Rita Cevera Juanes is presenting because she has a great track record of study in genetics, and you can sort of tell from her CV, which I won't go into detail, that she's made an excellent transition from having a robust training in genetics to looking at behavioral and neural aspects of genetics and epigenetics as well. She got her PhD from Polytechnical University of Valencia in 2004 under the advisement of Fernando Garcia Jimenez, where she was working with fibroblasts as a model for human ES cells. And then she went on to do a postdoc at the Salk Institute in San Diego after that, before moving to Oregon Health and Sciences University in 2006, which is exactly the time when I graduated. So we just missed each other. And she's and has been there ever since at the Oregon Primate Center. She's been research assistant professor there since 2016, and I won't give too much away about what she's been doing there, because she's going to talk about that. You can see from the title of her slide. She's been doing a lot of great work. Again, we're going to hear about it today, but she's been well funded to do this work. She has 2 R01s looking at the epigenetics of chronic alcohol use. And I'm super excited, as I know all of you are, to hear her talk about this work. So Dr. Cevera Juanes, take it away, please.
Thank you. Thank you so much, Paul, for the nice introduction. And thank you all for making time to attend this seminar today. And so I will be talking about the approaches that we use in our lab to identify neuroepigenetic signatures that are associated with long-term alcohol use, and how we try to use those signatures to identify functional targets that could be used as potential intervention for alcohol use disorder. Okay, so a little bit of background of why is it worth to look to study alcohol use disorder. I'm not sure how much familiarity there is in the audience for alcohol use disorder, so I'm going to briefly go through the main points of this disorder. So it's very prevalent, and there's 1/3 of the US population that meet criteria for AUD diagnosis in their lifetime. There's approximately 86% of individuals, adults over 18, that are currently alcohol drinkers in the US. And there's 6% of those individuals.
Oh, fuck yeah, of course. There's one.
You can hear me? Is there?
Yeah, we had some background noise. I muted him.
Oh, okay. Okay. And so among those, there's about 6% of individuals that are diagnosed with an IUD And a very small percent of those individuals actually receive treatment. AUD is highly comorbid with other mental illnesses and cancers as well.
Oh my God. I'm scared.
I muted him too. Everyone, just make sure that you have your microphones muted. Thank you.
Okay. And it's also becoming more and more prevalent among the elderly as well. And it is classified as the third leading preventable cause of death in the US. And it also has like a huge economic burden, like costing the US in 2010 approximately $249 billion. So the current treatments for AUD are scarce, and so they are pretty much limited to rehabilitation programs, so using psychological and social approaches. And then there's also a few FDA-approved drugs that are listed here, the sulfiram, naltrexone, and acamprosate. And so as you can see, there's not many options to treat this disorder. And unfortunately, there's about 2/3 of patients that relapse within months of completing treatment. And there's two factors, like one is that individuals with AUD, it's hard for these individuals to actually recognize that they have a treatment, and they don't seek treatment, maybe for that reason, or maybe because they are not too effective. And so there's a need for additional treatments. And this is basically what my lab has been funded recently to do, to identify potential novel targets for this disorder. So like I mentioned, there's about like 6% of individuals in the US that develop AUD. And so the question for me, or for in my lab, that we're interested in is in understanding why make these individuals different than the rest. And not only, but in particular, what are those molecular mechanisms that are mediating the transition from occasional and sporadic drinking to this loss of control over drug seeking and drug intake. And like I said, we're most interested in the molecular underpinnings of these adaptations that occur in the brain. And in particular, I'm interested in genetic and epigenetic regulation. And so what do I mean by this? So we know that there's a genetic component to AUD, but this is not explaining the whole heritability that we see in this disorder. And so there's something else. And so if we look at on the left side of this slide, to this plot A, the genetic risk is represented as this green bar that maintain, like staying pretty much stable through the lifetime of an individual, from birth to childhood into adulthood. And then through our life events, we go through experiences that can happen during childhood, but also in adulthood. And depending on these experiences, and it could be like environmental stressors or trauma that we go through, this could have an interaction with our genes. And depending on the degree of these interactions between our genes and these environmental exposures, there could be like our genes respond to that interaction in a manner that is reflected in a phenotype, that if it crosses a threshold as represented here, we may develop a disease. And so these interactions between the gene and the environment are mediated by what is the epigenome. And there's multiple types of epigenetic marks and modifications, DNA methylation, and then that's one that binds specifically to the DNA. But then there's others like histone modifications and small RNAs and other types of modifications. So mostly of my work has been centered on DNA methylation, and that's what I'm going to be talking about for the rest of the talk. And so pretty much, if we look at the plot on the right, the idea is that if we are in a more naive state, depending on the experiences that we go through our life, represented here in these different paths, and depending on what these environmental stressors are, whether they are negative or they are positive, we could have a shift toward a healthy state or a more diseased state. So what we're interested in understanding is what epigenetic signatures characterize the disease state and the health state, and see if we are in a disease state, how we can manipulate those signatures to revert into the state. So if we think more into what is the impact of this type of epigenetic regulation in the brain, and specifically in neuronal function, I'm trying to represent here what I could mean by this. So if this represents a neuron with dendritic spines receiving connections from other neurons, and this could represent an axon. And so under normal circumstances, this would be there would be a specific expression profile that allows this neuron to respond to the inputs that they are receiving. But in the presence of drugs of abuse, there could be signaling pathways that reach the nucleus, and then there could be a shift into the type of epigenetic signals that we have. So if we have a change in methylation, for example, in a particular gene that was being expressed, but now there's an increase in methylation in the promoter, and this gene becomes silent. So as a result of this, there could be alterations in protein function. And this could result in changes in the number of ion channels, in the conformation of the channels, and the different subunits that the channels are composed. There could be an increase in the, or a decrease in the reactive receptors, resulting in an increase in the neurotransmitter that is released to the synaptic cleft. So ultimately, there's alteration in circuitry, and this could be underlying the long-lasting adaptive changes in neuronal function that we see with chronic alcohol use. So this is one of the reasons why we're interested in investigating DNA methylation in particular. And so the overarching goal of our studies is, like I said, is to understand these neuroepigenetic signatures that may be underlying the neuroadaptations and behaviors that are associated with alcohol use. And the goal is to understand the biology and of this mechanism, and also to be able to provide additional treatments for this disorder. And so we're interested in different stages of the addiction cycle. So one is when we have alcohol naive individuals, we want to be able to identify risk. And so this is represented here by individuals that even before having consumed alcohol, they have an innate risk to become a future heavy drinker or not. Once alcohol is presented, and the individual actually makes the choice to consume small amounts of alcohol or heavy amounts of alcohol. And then if individuals that become alcohol use disorder subjects, if they at one point in their life, they decide to seek treatment, if they seek treatment, and whether we're also interested in understanding whether after receiving treatment, what happens with the epigenome with individuals that relapse, or individuals that become abstinent completely, or that they reduce their alcohol intake. So going back to that same figure that I showed in the previous slide, we want to add each one of these stages in alcohol naive, after chronic alcohol use, and post-abstinence. We're interested in understanding what are the epigenetic profiles of these non-heavy drinkers versus the heavy drinkers, and also how we could potentially revert that epigenetic signature that characterizes the disease state and towards a more healthy state. So how do we do this? So in our lab, we use a multi-species approach. Most of our studies use macaques, and we use tissue from the monkey alcohol tissue research resource. And we also use, in collaboration with the New South Wales Brain Tissue Resource Center in Australia, we use human postmortem tissue as well. And so the idea is to conduct a lot of our epigenetic and epigenomic and transcriptomic analysis with non-human primates and humans, but we're also including rodents, different strains of rodents, because we want to do a multi-species comparison of these profiles, epigenomic and transcriptomic profiles. We further use the rodents to conduct functional studies. So we select targets from our omic studies, and then we manipulate them in rodents. And then the idea is that when we find potential candidates that modify alcohol use behavior, we can test them back into a human primate, and ultimately they could potentially be used or considered to be studied for use in humans. So the model, the non-human primate model that we use for alcohol self-administration was established at the Primate Center by Dr. Kathy Grant. And so in this model, so at the Primate Center, we have a large pedigree rhesus macaque population, which is really important for epigenomic studies because we control environmental factors. And we know all their medical history and the relatedness with other members of the families. In this model, like this, the non-human primates have access to 4% ethanol, water, and food ad libitum for 22 hours a day for approximately 12 months and even longer, depending on the paradigm. So in this model, individuals go through a period of induction and that it lasts for about 3 months. And then after that, they have open access to alcohol for about 12 to 14 months. And here they have free choice to alcohol or water. And then at the end, so some of the cohorts will end after the chronic exposure, but then there's some of them that will go into 3 cycles of abstinence relapse. So overall, this model allowed us to investigate or to study individual variability. And this is kind of represented in this plot, where in the x-axis, we have the days in a year that animals are drinking. And then on the y-axis, we have the levels of alcohol that they consume. And we can see that there's individuals that classify as low drinkers, binge drinkers in green, and then in orange and red are the heavy and very heavy drinkers. So we could very nicely investigate individual variability and the effects of the dose of alcohol that is consumed. And then for those cohorts that go into abstinence, what is represented here is the amount of alcohol that they drink, equivalent after at the time of the chronic alcohol exposure, which would be equivalent to here. And then after each cycle of abstinence, the levels of alcohols, so this is representing the once they are exposed again at the beginning of the open access after one of these abstinence cycles, and then at the end. So we can see that there's actually an increase in alcohol consumption after the abstinence and not a decrease as we did. There's not really a decrease in alcohol. There's an increase at the beginning, and then the levels remain similar to the pre-abstinence exposure. So pretty much this allows us to study all the different time points that we're interested in the addiction cycles, the risk, the dependence, and then the abstinence. Okay, so using this model, I will be presenting data of the studies that we have done looking at the post-alcohol consumption. So after these 12, 14 months of chronic exposure, and this is summarizing the different types of groups, the different groups that we're going to be looking at. The controls, which are true alcohol-naive individuals, they never have access to alcohol. And then we use the non-heavy drinkers, represented as NHD. Those are individuals that consume less than 2 grams per kilogram per day. And then we have the heavy and very heavy drinkers, they combine together and they consume more than 3 grams per kilogram per day. So in our studies, we conduct pairwise comparisons across these 3 different groups: control versus non-heavy drinkers, and then control versus heavy drinkers. And we also compare what could be differentiating these 2 different classes of drinkers. Okay, and then very briefly, and to give an idea of what type of approach we use. Like I said, we mostly have focused on DNA methylation, and we use a genome-wide or an engagement approach. And so pretty much if we have 28 million CPGs in the genome, our approach targets approximately 3.7 million of those CPGs. And these CPGs are located in regulatory regions, in DMRs, in promoters, in DNA's hypersensitivity site, so they have regulatory functions. And so what we do is we share the DNA and we combine it with bioaccumulated RNA baits, and then we capture them using magnetic beads, and then we make a library and we sequence that in a HiSeq, and these days in an ANOVA-seq. And then the type of data that we obtain So it's kind of represented here. Like you can see, is a genome-wide, so it's an enrichment approach, and we only get reads, sequencing reads, whatever the dates are aligned in the genome and not in the whole sequence. And then we obtain differentially methylated cytosines, so individual sites. And this is shown here. These are 6 different CPGs, and this is representing the methylation rate. And we can see that, for example, in this case, we see a hypermethylated cytosine in heavy drinkers represented in red. Then we also do, we look for DMRs, which are stretches of differentially methylated cytosines that are close to each other in the genome. And so that's represented here. These are all different CPGs, and this is the level of methylation. And we can see that here there's about 10 or 12 of DMCs that are clustered together. So this is what we call a DMR, or differentially methylated region. Okay, so during my talk, I'm going to, so we look at the changes in DNA methylation in different brain regions. And as you probably are all familiar with this, there's different brain regions that contribute to different aspects of the addiction cycle. And so 2 of the brain regions that we look at is the nucleus accumbens, which is located somewhere here. And this brain region is involved in motivation, regulating motivation and rewarding behaviors. And that's one of the brain regions that I will be presenting data on. And the other one that we started to look at is the dorsolateral prefrontal cortex, which is somewhere here. And that is involved in executive function. And I won't present the data exclusively from one brain region to another, but I will highlight when I'm talking about one and the other. And so independently of the brain region, the type of analysis that we do has been giving us the same type of results. And so one of the first findings is that when we compare across the different groups, the control, the non-heavy drinkers and the heavy drinkers, we in general see that there's that with chronic heavy drinking, there's an association with hypermethylation. And this is shown here in these heat maps. When we include heavy drinking in the comparison, either versus control or versus non-heavy drinkers, there's a tendency to have more hypermethylation sites as compared to non-heavy doses, like low doses of alcohol present. Another thing that we saw is that by comparing these 3 groups, we can identify sites or regions. In this case, these are DMRs. That show a different type of response to the dose of alcohol. So we call some of them alcohol responsive because independently of the dose of alcohol that is consumed, the methylation level in non-heavy drinkers and in heavy drinkers is different as that we detect in the controls. And then we have those that are alcohol correlated. The more alcohol we consume, the higher level of methylation we have, or the lower level, but they are correlated in a dose-dependent manner. And then we have a subset of those that we call low dose specific because the change in methylation is unique to the non-heavy drinkers, while controls and heavy drinkers have a more similar level of methylation. And lastly, we have the heavy dose specific, which they are particularly high or low and exclusively in the heavy drinkers and not in the other 2 groups. Another finding from these studies that we see is that a lot of our DMRs are located within the gene. So there's some of them in the promoter, but mostly they are intragenic, and they could be located in either introns or exons. But our preliminary data on this suggested that it could be regulating alternative promoter use or alternative transcript use. And so this is an example. Oops, sorry. This is an example of one of these DMRs. This is one gene, JAK-MYC1. It has 2 alternative exon 1s, exon 1A and exon 1B. And then it has the DMR is located within this exon here. And so our methylation data show hypermethylation in heavy drinkers as compared to the non-heavy drinkers, shown in here. And when we looked at the expression, either using exon 1A or exon 1B, we saw differential expression. So exon 1A was downregulated, but 1B was upregulated. So this was suggesting that there could be that the DNA methylation could actually be contributing to regulating the use of one promoter, one exon or the other. And lastly, a lot of the DMCs and DMRs that we identify in our studies map to genes that have a relevant or potential relevant role in alcohol use disorder. And I'm just listing here some of them which are encoding for receptors or for trafficking, receptor trafficking molecules. And I will talk about a couple of those in a second. Okay, so up to now, I showed data that, okay, so we run DNA methylation analysis with signals. We're picking up some interesting effects of this DNA methylation, the dose effect, the regulation of alternative transcripts. But the question for us was how functional are these sites or these regions? And so I'm gonna show an example of 3 of these, actually 4 of these regions that are associated to genes, and how we're using different approaches to manipulate the levels of this of the protein that is encoded by these genes in the first example that I will give, which is the GPR39. And then we're also manipulating the specific transcripts for other of these genes, like MBA and one of the orexins. And lastly, I'll show an example of one of the genes where we're actually using epigenomic editing tools to manipulate the DMR and see effects on expression. Okay, so we're starting with the GPR39. So GPR39 is a sin-sensing G protein pathway receptor, and it's known to be downregulated in the brains of suicide victims. There has been models of using GPR39 knockout mice that showed that these animals have depression and anxiety-like behaviors. There are another study that identified or suggested that GPR39 modulates the presynaptic glutamate release via the endocannabinoid pathway. And others have found a relationship between GPR39 and BDNF and CREP, and also in regulating GABA function through the PCC2 cotransporter. And so our data in the non-human primate showed that there was a hypermethylation in heavy drinkers, as shown in this plot here. In red, they are heavy drinkers, in blue, not heavy drinkers. And so there's an increase in methylation, and that was associated with a decrease in expression for this gene in heavy drinkers, and this was negatively correlated. And so we hypothesized that if we could use an agonist for GPR39, maybe we could be able to reduce alcohol intake. And so what we did was to use rodents to test this hypothesis. And we used the TCG 1000 inhibitor is an agonist, it's not an inhibitor, it's an agonist for GPR39. And so we first did a dose response using, like I said, black 6 mice. And so we allow first the animals to escalate alcohol intake during 16, 17 sessions. And then after that, we tested different doses of these agonists. And our results showed that when we used a 7.5 milligram per kilogram, we were able to reduce ethanol intake and preference without altering total fluid intake. And so we used this dose, and we ran locomotor activity and a saccharin preference test to see if there was any underlying reason why this reduction in alcohol was, and it wasn't specific of alcohol, but we didn't see effects on locomotor activity or effects on saccharin intake. So it indicated that the effect might be specific of alcohol. And then we used that same dose to run an intermittent access to battle choice model. So in this case, the animals had access every other day to alcohol for 24 hours, 22 to 24 hours. And we first started by separating the animals into 2 groups. One set of animals would receive a placebo injection and the other the agonist. And that could happen prior to the ethanol session. So we saw that those that received the agonist did not consume any alcohol, pretty much, very low doses and pretty much no alcohol, while those that received the placebo, they were increasing, escalating the intake. And then what we did after this session was to remove the agonist. We just injected the placebo. And we saw that there was an increase in escalation in alcohol intake in these individuals as well. And then at this last session, what we did was to administer an acute dose. And then we saw that there was again a reduction, an acute reduction in the alcohol intake. We looked at whether there was any changes in BDNF, ACC2, and in the nucleus accumbens, which was one of the areas that we were interested in looking. And we saw an increase in BDNF expression and no changes in ACC. We also looked at excitatory and inhibitory currents in the nucleus accumbens after using different doses of the agonist that were added to the ex vivo slices prior to recording. And we saw that there was an increase in the excitatory frequency in the presence of the agonist, of different doses of the agonist. That's indicating that the agonist may have an effect on median spiny neurons in the nucleus accumbens. So the next obvious question for us is whether we can go ahead and test this agonist in human primates. However, before doing that, I'm very interested in understanding how is GPR39 actually modulating, excuse me, modulating alcohol intake. And so to do that, there's a lot of studies, of molecular studies that I would like to conduct in mice prior to jumping into testing anything in the human primate. And so one of the first things that we did after that was to see, okay, we know that the GPR39 levels in human primates, as shown here, were lower in the heavy drinkers as compared to the non-heavy drinkers. And then we went ahead and used this agonist with the mice. But in reality, we didn't know what is the response in mice of GPR39 with alcohol. And so we ran this study using GPR39 knockout males and females. And we did an intermittent access on these individuals. We had wild types and knockouts. And our results show that this is the ethanol intake for males, this is for females, and the blue are the wild type individuals and the orange are the knockout individuals. So indicating that those individuals that do not have the GPR39 show increased levels of alcohol consumption, so both in males and females, and most prevalently in the females. We didn't have female data from the human primates, so this was a good thing to see in the mice, and that the, that the behavior is very similar to what we saw in the non-human primates. The next question is how how is actually GPR39 affecting the alcohol behavior that we see? Is that a direct effect in the brain or is indirect? Because GPR39, as you can see in this is a figure from a review from Dr. Rita Cevera. And what they're showing that GPR39 is expressed in multiple tissues in the body, and that using the agonist, there's effects, like people have been conducting studies in using multiple different organs. So our question that we don't have the answer yet is whether the effects that we're seeing in behavior are a direct effect of this agonist in the brain, or if it's mediated through something else. For example, they showed in a study that there's an effect of the GPR agonist in the gut, and we know that there's a direct relationship between the gut and the brain, and there's alteration in somatostatin and ghrelin. So that could be a possibility that the effects we're seeing is through the gut and not exactly in the brain. So we and others have shown that there's GPR39 expression in the brain. And this study looked at multiple GPRs in different brain regions as shown here. And I'm just highlighting that the one that we were looking at, which was the accumbens, there's expression in the accumbens, but there's also expression in other brain regions as well. And in terms of what cell types could be expressing this, so there was this in the Dropwith's website, it's suggesting that median spike neurons may be expressing GPR39. And we have conducted some single nuclear agonistic in non-human primates. And our data, however, shows that there's more of the expression of GPR39 in microglia as compared to any other of the cell types. So there's a little bit of unknown of what specific cell types might be expressing it, but definitely there's expression in the brain, which was one of our first questions. And then we also wanted to know if the agonist that we used was actually reaching the brain. It was crossing the blood-brain barrier. And so we know from our prior study when we did the dose response using the different doses of the agonist that during the first 4 hours of, so when we administered the agonist and then we let the individuals consume alcohol, the most effect of reducing alcohol use was seen prior to 3 hours. This was the 7.5 dose that we used. And then after 3 hours, it seems that it's recovering, the ethanol intake goes back up. And so what we did was to use the wild types and the knockout animals that we had, and we injected the agonist and then we collected the brains 1 and 3 hours after this injection of the agonist. And then we measured the levels of the agonist in the brain. And so our data showed that after 1 hour, either in females and in males, and this is a very small study, so there's only 2 individuals per group and 1 individual free only, so very preliminary. But when there was so after 1 hour of administering the agonist, we were able to detect levels in the brain, both in females and males, and nothing when we administered the placebo. And then after 3 hours, we can barely detect any of the agonists in the brain for any of males, in either males or females. So then indicating that yes, it looks like the agonist is able to reach the brain. And so going back to our question of how is GPR39 contributing to the modulation of alcohol intake, the studies that we're conducting are leading us towards maybe being able to answer this question in the near future, but we have the ability to use a knockout model that can help us answer a lot of these questions. We know that there's GPR39 expressed in the brain, and we know that the agonist is able to reach the brain as well. We're actually conducting single nuclei agonistic analysis to determine in which specific cell types GPR39 is expressed in different brain regions, because this will allow us to potentially design viral approaches to specifically target GPR39 in particular brain regions and see how those affect the drinking behavior. We're also testing additional agonists in collaboration with Dr. Holz from the University of Copenhagen. We have access to this new agonist that is independent on the presence of zinc. And we're also going to test that to see if it reaches the brain and how long the effect lasts in this case. And then we're also working in collaboration with Dr. Anthony Barnes at OHSU, who has a lot of expertise in understanding the signaling cascades that are activated by GPR39. But we're specifically interested in the context of alcohol in the specific brain regions. And then another aspect we're very interested in is in identifying individual variability in the response to this agonist. And this graph here shows, for example, that in the same study when we administered the agonist for the first sessions and there was no alcohol escalation in these individuals, and then we removed it and there was escalation, there was a subgroup of those individuals that never escalated to consume. So we're interested in understanding what's different between these individuals and what could be protected for alcohol escalation as well. So overall, in my opinion, all these prior studies will bring us to a point where we will be more confident in being able to test this agonist or another agonist in non-human primates. Okay, and then I'll move on into a different approach that we use, and in this case, when we're particularly modifying specific transcripts. I will give a brief example of MBA, which this gene was identified when we were comparing DNA methylation data sets from non-human primates and humans. And this is represented here. We had, in humans, we don't have all these subclasses or drinkers, so it's AUD versus controls. And we compare them to the different groups that we had, different comparisons that we can do in the non-human primates. And among the genes that were in common between them, there's an example here of some of them, but one of them was MBA. And MBA encodes for neuroepigene. And neuroepigene is a transporter of glutamate and GABA receptor subunits from the endoplasmic reticulum to the membrane. And it's also involved in the recycling of these subunits into the membrane. And so our data, both in rhesus and in human primates and in humans, identify differentially methylated regions pretty much in the same genomic region. And the changes in methylation were in the same direction. So in the human primates, we have hypermethylation in heavy drinkers. And then in the humans, we have hypermethylation as well in the AUD individuals as compared to controls. And so based on the location of the DMR, which is intragenic, as you can see here, we are looking into exome usage analysis. And we did this in the human data set where we had enough depth that we could look at exome usage analysis in the RNA sequencing. And our data shows that there's some of the exons that show differential expression between AUDs and control. And so the current approach that we're taking is into, so this is suggesting that the shorter transcripts may be differentially expressed. And so our current approach, we're using viral tools to design, to overexpress these shorter variants in this case, in the preventive area of rodents and CDFF. So this is one of those examples where we found a common signal between human primates and humans, and we're using rodents as well to functionally validate the potential role of this gene in AUD. And then another example is the neurexins. So we identified this gene in this genome-wide DNA methylation analysis we did in the dorsolateral prefrontal cortex in a human primates. And when we did a pathway analysis of all these differentially methylated signals and the genes that they were mapped by, we identified several neurexins within the synaptogenesis pathways that was altered both in males and females. And so neurexins are considered hubs for cell adhesion, and they have, if you look here, neurexins are located in the presynaptic side of the synapse, and they interact with multiple different types of postsynaptic molecules. And there's different types of neurexins, 1, 2, and 3, and each one has alpha, beta, and sometimes gamma transcripts or isoforms. And so, and they depending on whether we have alphas, betas, or gammas, so they have different extracellular domains. And these different domains determine which kind of partners they're going to have in the postsynaptic level. So pretty much they have a very potent role in modulating synaptic plasticity depending on what's expressed, the alphas, the betas, and what they interact with postsynaptically. So our data, like I said, we identified different neurexins, but in the case of males, we saw differential methylation for neurexin 1. In non-human in rhesus. And so this is where the DMR was located. And this short transcript here, it's the neurexin gamma. This could be the beta, and this could be the alphas. And so our data show hypermethylation. And then we looked, this is very busy, you don't have to look at all of this, but this is because we look at the expression of all the different transcripts. And we saw that there was a decrease in the expression in heavy drinkers for the neurexin gamma, which is the short one. And then in females, however, we saw a change in expression for the beta, which could be this one here. In the rodents, we then look at what the expression levels would be in males, in this case in rodents. And again, we saw that there was a decrease in the expression of the neurexin gamma. So we have designed a virus to modulate the expression of this particular transcript. And we're in the process of injecting that in the prelimbic area of rodents and look at the effect. What we have done so far has been more, we have done a little bit more work with neurexin 3. And in this case, this was particularly differentially methylated in female heavy drinkers. And this is where the DMR is located. And we see that there's a hypermethylation in heavy drinkers as compared to the other groups. And our expression data, showed that there was a decrease in expression in heavy drinkers. So again, we actually have designed a viral approach to overexpress the expression of this particular transcript. So this is some of the in vitro work that we did. We used this lentiviral construct to overexpress neurexin 3 beta. We transfected NK cells. We did FACS sorting, separated those that were GFP positive from those that were negative. We confirmed the expression of the HA tag that is linked to the neurolepsin 3 beta in the vector backbone. And then we looked at the expression levels to confirm that there was actually the expressing neurolepsin 3 beta. And we see an increase in expression of the neurolepsin and similar expression of GFP in both the negative and the positive cell. And then we used this construct and infused it in the prelimbic area of mice, and we exposed them to intermittent access to bottle spirogram of alcohol intake. And what we saw is that during the first sessions, from session 1 to about session 7, there was actually a reduced consumption in those individuals that were expressing neurolepsin 3 beta. However, this effect disappeared in the following session. So we don't know why this is for, and it could be because there's some kind of compensation with other neurolepsins or not, but this is something that we are going to look into. And lastly, I would like to give a brief, okay, I don't have much time, so I'll give a very brief overview of how we're using epigenomic editing tools to alter the expression of other DMRs. In this case, I'll talk a little bit about ILK. So ILK again was identified in the dorsolateral prefrontal cortex of non-human primates. And in this case, we only saw this enriched in males. And so ILK has been linked to plasticity, memory, and cognitive deficits, both in a model of fetal alcohol syndrome, and also has been identified in having a role in cocaine addiction as well. And so again, looking at what our methylation data showed, this is what the ILK, the different transcripts for ILK. This is exon 1 and it goes in this direction. And this is another gene, RRP8. And so the DMR is located at the promoter level of both of these genes, and it was hypermethylated in drinkers. There was a study done a few years ago where they saw that what they characterized the promoter of this gene, and our DMR actually overlaps with the promoter, as we can see here as well. And so what we did was first to look if only restricting the region to our DMR if it had a promoted activity. And our results showed that yes, in fact, it has promoted activity. And then what we did was to look at the expression of either of these two genes because we don't know exactly what could be regulating. So we look at the expression of ILK and also of RRP8, and we only saw an effect on expression for the ILK gene. What we did next was to use a DCAS9 VPR approach. VPR is a transcriptional activator. And so we use guide RNAs. So we use the approach described in this paper. And we used different guides overlapping with the DMR. And then we transfected that with the DCAS9 VPR vector and to mainly T cells. And we saw that some of these guide RNAs are shown here were able to increase the expression of ILK. So what we are doing now is actually being more like refining our approach and using this recent tool that has been recently developed to be able to persistently manipulate the methylation levels of the DMR and look at how we can actually, if these DNA methylation levels actually have a role in altering the expression of the ILK gene. So overall, from what I have presented, I hope that I told you that there's using the integration of our epigenomic and transcriptomic approach, we have been able to identify those effects, individual variability, potential role of the epigenome in regulating transcript, the expression of different transcripts. And this has given us this integrated framework that allows us to investigate, but also to manipulate the specific targets and see what the effect is on modulating alcohol intake. And so by doing this across multiple species, we are able to, like I mentioned, use exploiting this potential role of the epigenome in regulating transcript expression. And and investigating how that could be underlying the synaptic plasticity that we see in these models. By also looking at multiple brain regions and including cell type specific atomic signatures and integrating all together, we are working towards being able to identify manipulating not only genes, but specific regulatory regions that either by modulating the gene expression as a whole or a specific transcript allow us to alter or modulate behavior. So with that, I would like to thank everybody in my lab as shown here, especially Carlos and Priscilla, Lara, and Noah. They have done a lot of good informatics analysis. And then my collaborators at OHSU, at the Brandon Center and also at OHSU, and of course to my funding sources. And with that, I thank you all for sticking around, and I will be happy to answer questions.
Thank you, Dr. Cevera Juanes.
Mm-hmm.
I hear the applause rippling from around the world.
Oh, thank you. I can't see anything. I don't know why. Sure. Okay. Now.
I have some questions, but before I ask my long-winded questions, I'd like to open it up to the rest of the attendees. Either you can unmute yourself, ask your question yourself, or if you want to put it in the chat, that's fine. Does anybody have any questions? I'll start. I can break the ice. Okay. So I loved your talk. I think what you do is so important and unique. You're really well situated to do what you do. One, because you're clearly a talented scientist, and two, because you have this ability to take this multi-species approach. And I think that that's really important, especially for assessing individual vulnerability, because you can think about people with alcohol use disorder drinking for different reasons. And that's some of that. You talked about variability in terms of like how much people would drink or monkeys would drink. But I wonder more about like categorical distinctions between the reasons why someone might have alcohol use disorder. And so like, I think of the slide you showed with the brain areas, you talked about, Koob's negative reinforcement, but you also mentioned positive reinforcement, loss of control, and it like the sort of downward cycle. But those could be coexisting categories where different people have problematic alcohol drinking for those different reasons. So I'm sorry, this is a long-winded question. I remember a couple of decades ago, Kathy Grant, when she was at Wake Forest, was doing studies with monkeys looking at discrimination between alcohol and GABA drugs and NMDA receptor antagonist drugs. And sort of she demonstrated that there were some monkeys that appeared to be drinking for the GABA agonist, the GABA positive modulating stimulus effects of alcohol, whereas some monkeys would drink for the NMDA antagonist properties of alcohol. And so my long-winded question is, do you ever, how would you approach taking the epigenomic modifications and applying that potentially to the categorical individual variability in your, in particular, your primate subjects? So
really, really good point. And so my short answer would be size. So for omic studies, we need size, right? And so one of the limitations for me right now to answer that question could be, I couldn't have enough individuals to look at that. However, they are like more and more, as Kathy is increasing, more and more we have more individuals, and they are including a lot more behavior studies in the monkeys. And so we might be able, I would say that right now the sample size would not allow to make that very particular, very defined distinction. But it could be something that with integrating their cognitive behavior studies that they are doing, and a lot of the other, in my mind, my goal could be to have these profiles from multiple brain regions. So integrating all that is going to possibly provide a lot of that specific detail of how different brain regions are connecting to each other and how that could be distinguished in these categories that you talk about.
That's great. Thank you. Thank you. Other questions? I do. Since no one's asking questions, I'll use my prerogative as the host. Yeah. So you mentioned multiple brain areas, and so I imagine when you're trying to focus, trying to drill down to targets like GPR 39 or other targets, that you're looking at the epigenomic modifications and then trying to find where that leads to a difference in expression in a particular brain area. Do you ever feel like that narrows it down too much, right? Because just because you might be looking at a certain brain area, that doesn't mean, and you don't see an expression difference, for example, that doesn't mean that those modifications might be leading to expression differences somewhere else in the brain or somewhere else in the body. And so I wonder, how do you think there are missed opportunities, and how often do you think that that's happening where you might ignore an important target? I'm just sort of curious as to what you think about when you choose candidates to go after.
Yes, you're absolutely right. And I think about this a lot because one of the goals for integrating different brain regions is exactly that, to see what is common, what is different between brain regions that might be directly connected through, projections and stuff. So the limitation, and that's why I'm taking this approach right now, is that I don't have the ability or the financial support to do this type of analysis from multiple brain regions. Right. And so I have to work with what I have. And it's limited. So right now it's limited to right now I have 2 different brain regions at least. So I'm starting to integrate that, and not only limiting to DNA methylation, I'm including other epigenetic marks as well. And that, I think that is going to be a factor as well, when I'm not limiting myself to DNA methylation only, but including other, these other, like chromatin conformation, and then the ataxic data sets, all of that. It's going to help us better refine what regulatory regions, what type of regulation these DMRs may be doing, or other open access chromatin areas may be doing. But ideally, it could be to eventually, like in a perfect world, it could be to be able to have enough funds to characterize all these different brain regions, and specifically at the single cell level, it could be just the best of everything.
Well, if it was up to me, I would certainly send more money your way to do that.
Good.
Other questions? All right, let me see if I can scare even more people away with more questions. I just have one more, if you'll permit me.
Oh, yeah.
So I thought, for someone that does, genomic studies and then focuses in on a candidate like GPR 39, you just had such a great story to tell, and it was really exciting. Like those are the genes and the molecules, that as someone who does mapping studies, like I'm trying to find those stories as well that I can tell. I guess like in terms of like a career strategy, or a research strategy, like when do you decide that you want to spend more of your efforts following up candidate genes like GPR 39 versus doing more mapping studies. I'm just sort of curious when you start to put your chips down on the roulette table on particular candidates, as opposed to spinning the wheel again.
Yeah, it's so
a bad analogy, but
that's a hard question actually to answer, because so to me, for GPR39, it was pretty clear, because the effect, there's an obvious effect, and it was something like, yeah, like we have to follow up on this because there's an effect in there, and so to me, like leaving it there, like we used an agonist and there's a reduction in intake, and just abandon the story there, it doesn't make sense to me, like professionally, I guess, because I think there's a lot more to know about how is that molecule doing anything, and how can we exploit that for the ultimate goal of providing a treatment. So that's what drives me is that. And then, of course, I do omics, and I get plenty, I get like hundreds and thousands of hits. And I have a lot more to look at, right? From all these data sets. And so, first of all, it's hard to actually decide what you're going to go after, first of all, from all these genes and all this stuff. But once you find something that is promising, I stick with it until I know, until I can help figure out how that mechanism is working and whether it could actually lead to a treatment or not. So that's my personal take. But I totally get it, because we could, there's a sea of targets out there.
That's the question that I like to ask everybody. It's like each of these molecules could spawn a research program. So what do how do you decide to grab hold of one?
Yeah. And then the other, I could add one more thing too, is to know, it helps me also to know where in the pathway that molecule is located. Like, could it be a driver of the behavior, or it could be a consequence of it? So that's something that helps me know when to follow and when to not follow so much.
Well, thank you so much for answering my questions. Sorry, I monopolized everything. Hopefully I didn't steal anyone else's time. So unless David or Catherine or Anna have questions, we'll end it because we're going past the hour. I just want to say it was a great talk. It was a great way to start off our seminar series. So I appreciate that immensely. And just thank you so much. I really appreciate it. And it was a pleasure to meet you.
Yes, likewise. It's been a real pleasure. And I wasn't a member of the IBANGS before, but I'm going to be now.
That is good, because the whole reason I'm doing this is because I'm part of the membership committee of IBANGS. And so we're looking for members. So if you join, then you've validated my existence.
Perfect. I'm going to do it today. So
all right, great. Thank you very much.
Thank you so much. It's been a pleasure. Thank you.
Bye-bye. Goodbye, everybody.